Ujú. Buenas tardes a todos. Esperemos que todos se encuentren muy bien de salud y no bajando la guardia ante esta pandemia. Muchas gracias por acompañarnos el día de hoy y en representación de la Asociación Mexicana de Nutrición Animal, les doy la más cordial bienvenida a un nuevo seminario de nutrición de este 2021. En esta ocasión se lo estamos dedicando a la nutrición de las mascotas. Y para esto, un gran amigo de Amena, el doctor Shiguang Yu, a quien mucho agradecemos su participación, nos hablará del gran impacto que tiene el microbioma intestinal en la salud de la mascota. Este evento está patrocinado por la compañía ADM Animal Nutrition, a quien también agradecemos su apoyo. Les recuerdo que va a ser en inglés, sin embargo, en las herramientas ustedes podrán encontrar en la parte posterior, en interpretación, podrán encontrar un canal en donde viene la traducción simultánea al español. El evento será moderado por nuestro experto en el área de mascotas, el doctor Miguel Ángel López, a quien dejo el micrófono y te agradezco mucho también, Miguel.
Muchísimas gracias. Gracias a todos los miembros de Amena por haberme invitado a esta presentación. Para mí es un honor siempre participar con ustedes y para mí es mucho más honor también estar aquí conviviendo y presentando y trabajando junto con el doctor Yu, que es antes que nada un gran amigo que conozco desde hace muchos años y que le he aprendido muchísimas cosas. Entonces, la verdad es que también creo muy importante y muy interesante que participen ustedes en este webinar y en esta presentación que va a dar el doctor Yu. Para leer un poquito y platicarles un poco acerca del background y de la carrera del doctor Shiguang Yu, les quiero decir que tiene una maestría y un doctorado, y su doctorado viene de la Universidad de Wageningen de Holanda, es su grado doctorado, y su maestría de la Universidad de Baker de Estados Unidos. Y desde que se graduó de su doctorado, él ha trabajado en diferentes universidades, como la de Utrecht, como la Universidad de Holanda, como la Universidad de Davis en California, como un asociado investigador. Así también ha hecho estudios y trabajos de postdoctorado e investigación, y además ha hecho muchas investigaciones y trabajos acerca de nutrición de mascotas. Algunas de sus investigaciones y su trabajo de investigación es tan importante que ha sido citado en el NRC, en el mismísimo NRC, citan los trabajos del doctor Shiguang Yu. Tiene una gran experiencia trabajando en empresas líderes de nutrición de mascotas, principalmente en el área de nutrición, investigación y desarrollo, nuevas tecnologías y desarrollo, así como formulación y diseño de productos. También en el área de ingredientes, desarrollo de ingredientes, calificación y calidad de ingredientes, e incluso mercadotecnia y apoyo a los clientes. Él ha emitido y ha aplicado y tiene múltiples patentes y ha sido autor de muchas publicaciones científicas publicadas en varios papers y obviamente específicamente en nutrición de mascotas. Él es actualmente un investigador en el área de animales de compañía, en el área específicamente de nutrición e investigación de la empresa, como ya se mencionó, ADM. Pues una vez más es un honor tenerlo aquí, doctor Shiguang Yu, y una vez más un honor estar aquí moderando su trabajo. Lo dejamos con nuestros participantes. Muchas gracias.
Good afternoon, everyone. So today, let me minimize this one. So today I'd like to discuss with you gut microbiome and pet health. So I'll talk about the microbiome. We're not talking about a very deep technical part of microbiome. Rather, we're going to talk about it from a high-level view that is relevant to pet food and the pet food industry, pet nutrition. With that said, I'm going to cover 4 areas. One is microbiome market and research. And then we'll talk about microbiome as a general. Then we'll focus on the microbiome, gut microbiome, focus on the gut microbiome because it is most relevant to pet food and pet nutrition. Then we'll talk about manipulation of gut microbiome because we are talking about pet food and we're talking about the gut microbiome. And we like to use food to make a microbiome more healthy, so can promote the healthy of pets. With that, and then we'll focus on more on the prebiotics and the probiotics and the postbiotics. So that's kind of areas I'd like to share and discuss with you today. So first to talk about the microbiome market trend. And this is study was made by Market and the Market. And in this report, you can see this is a human microbiome market trend. Now we are in 2021. So this report predicted by 2027, human microbiome market, food and the beverage supplement. By 2027, it could reach $1.7 billion with CAGR of 22.5%. So that's a significant growth speed. So this is the human market trend. As we all know, pet food is very closely following the human health in the market trend. So because of the human significant growth in the microbiome market, we anticipate, or I anticipate that the pet food industry will follow. We probably will also see this microbiome become a new innovation areas for the pet food industry. So this is the on the commercial part. What about the research part? You can see this study published in 2019 to show the number of publications in microbiome areas. The top panel is the number of publications in prebiotics and the probiotics from 2000 to 2018. You can see significant number of publications in this areas, and that's include the humans, animals. So this is a significant increase from 2000, about 200 publications to 2018 is 3,667 publications. And then on the bottom one is called the probiotics. We'll talk about more about this probiotics, and you can see this emerging trend in the research areas, and then it's about 1 or 2 publications a year. In 2018, it jumped to 25. It's still very, very few publications, but this area is very active. So it could be an emerging trend in the next 5 or 10 years to become a major activity, and it gave us an opportunity to do the pet food innovation. Now, what is microbiome? So you will probably read the publications. You will see various definitions, but they are very close, all very close. And the last year, the expert in this area, so they gathered together and they published this paper. In this paper, they call the revisit microbiome definitions. So that's come up with this nice summary. What is a microbiome? If you look at this slide, I took this from the paper, and you can see there's 2 big pieces composed this microbiome. One is called the microbiota. So this basically talk about all the microorganisms. Include bacteria, archaea, fungi, protists, and also algae. The other big part is the theater of the activity. So where those microorganisms work and what's the results of those things. There's 4 major components in the theater of activities, and one is the microbial structure. So those components build the microorganism, proteins, lipid, polysaccharide. The other parts, they are metabolite, those microorganism metabolite. You can see the signaling molecules, the toxins. So a lot of things there is part of the microbiome also. The third part is called the mobile genetic element. In that microbiome big environment, it's virus, phages, and also small piece of DNA. So they are moving around between those microorganisms. So the fourth part is the environmental conditions. Basically, it's kind of a habitat those microorganisms live in. So it depends on which habitat. In our case, we talk about animals, we talk about humans, we talk about bodies, organs, those things. So that's also confirmed part of the microbiome. So you can see it's very complex. But in reality, if you look at all those publications, for the microbiota part, most of the research is doing on bacteria. Very few research doing other microorganisms. And in the environment part, it's majorly different organ or tissues. And then we'll talk about in the next slide. So that's why I talk about the habitat areas. And then you can see if it's talking about the gut, it's the microbiome, gut microbiome, and the skin microbiome, lung microbiome, and urogenital areas of microbiome, mouth, nose, ear, different part of the body, different organs that can form the different microbiome. And very interesting, I also saw the publication talk about the blood microbiome. As everyone knows that the blood is sterile, there's no bacteria, but they do, using the high throughput technology, found the bacterial genes in the blood, and they talk about the blood microbiome. But anyway, I think gut microbiome is mostly researched areas, and it's very, very relevant to our business, pet nutrition and the pet food. So today we'll focus on the gut microbiome. And this is why we're focused on the gut microbiome. You probably heard a lot of this gut-brain axis, gut-kidney axis, gut-skin axis, gut-lung axis. Gut microbiome has a great relationship with the different organs. Some people, scientists nowadays, even call gut microbiome is another organ of the part of the body. So demonstrate it's important for the health and for the animal life and also human life. There's a lot of research found the relationship or association between microbiome and the disease conditions. For example, IBDs, allergies, atopic dermatitis, overweight, diabetes, metabolic syndromes. So a lot of diseases have some kind of relationship with the microbiome in the gut. And also the normal physiologies. Microbiome can also affect the digestions, performance. If you go to the human side, you will see a lot of sports nutrition. They talk about the gut microbiome affected the sportsmen's athletes, the performance in their sport competition. So microbiomes are very, very interesting, and they have a lot of effect on different areas of disease, normal conditions. That's why it's interesting for us. So how do you assess the gut microbiomes? And you talk about the gut microbiomes. How do you evaluate it? How do you assess is it good? Is it bad? Traditionally, you use those culture to do the culture plating to culture the bacteria. And here again, we talk about the bacteria because other microorganisms is difficult to culture. And even the bacteria, because the gut bacteria live in a very unique environment. A lot of those bacteria is not culturable, so you cannot culture them. With the new technology development these days, this high throughput technology really help us to expand into this microbiome research. Most popular used or commonly used high throughput technology in the microbiome research is called the 16S RNA gene sequences. And you can see on the right up here, and this is the 16S RNA. You can see the gene consists of constant regions, also various regions. So this various regions gave us the opportunity to identify unique genes, and that we can use to identify the bacteria. And that's the basic of the 16S RNA technologies. How to do it? Typically, if you look at the left lower side, that's the way you typically do it. You get the sample, extract the DNA from the sample, and then do the 16S gene sequence using PCR. And then further do the data analysis. You can identify the genes that will be used to identify the microorganism. So for the gut microbiomes, most often used the sample is the fecal. It's a fresh fecal sample because it's easy to access. And then when we do this kind of research, like I mentioned in the microbiotas, and we typically or often measure the bacteria. It's not other microorganisms, but it will be nice to know the whole picture, all the other microorganisms. But if you look at the publications, most often we measure is bacteria because that's the predominant microorganism in the gut, in our gut. So when you measure the microbiome, you have all this 16S, you know, all the bacteria and microorganisms. Now, how do you evaluate it? I mean, what are you looking at? So what we're typically looking at is the composition of the microorganism, what kind of microorganism in that samples, and also the abundance of each microorganism. And then also look at the diversity, how many different kinds, and each of them is just a lot of one bacteria, or different kind of bacteria also have different large quantities in there. So the more diverse of this microorganism in the microbiome samples, and typically that means the better, more healthy. The figures on the right lower part, you can see is an example of research, and they look at the dog gut microbiomes, the dog with IBDs and the healthy dog. So if you look at those bacteria, the relevant, the relative abundance of bacteria in this two group of dog populations, you can clearly see the composition is different, the abundance is different, and the diversity is different. So gut microbiome really associated with different disease and different conditions. Now we talk about the gut microbiome, taking a dog as an example. So gut, you know, is talking about from mouse to the large intestine to the rectals. There's several components, compartment in the GI system, from mouse, stomach, small intestine, and the large intestine. And each segment or each compartment have very different conditions. For example, in the stomach, the pH is very, very low. Because of these different conditions, the microorganisms in those compartments are also very, very different. And if you look at the pie chart, this study published in 2016 looked at, different sections, 3 different sections of intestine, and then look at the bacteria and microbiome. You can see the composition is very, very different at each section. We all know most of the bacteria are located in the large intestine. So that's the major interesting part when we're doing the research in gut microbiome. As I mentioned, when we do the gut microbiomes, you see most of the sample is a fecal sample, fresh fecal sample. And there's limitations using fecal sample to predict or reflect gut microbiome. I'll talk about that later. And also, as I mentioned in the earlier slide, oftentimes measuring bacteria, not the other microorganisms. So keep those in mind. There's some limitations. We're looking at part of the picture of the gut microbiome by this kind of methodology. So here's an example of composition versus a function when we talk about the gut microbiome. Composition is very simple. If you look at the upper panel, that's the microbiome of a phyla, a different section of the GI system. And you can see the bacteria are very different at each different section. But if you look at the lower part of the panel, it's a metabolomic pathway. At each different sections, you can see that metabolic pathway are very similar. So it's not like the phyla composition. So that's another thing, is a new development when you do the gut microbiome research or studies. You cannot just look at the composition. You have to look at the metabolic pathway, the function part of the gut microbiome. You may have different bacteria composition in the samples, but they could have a similar metabolic pathway. So that the difference in the composition may not matter, but the functional metabolically pathway or functional gut microbiome is more important. And you can see here is a very, very similar situations at different section of the samples. And then here's the composition at the phyla level. You can see it's a very, very dramatic difference. This is the stools. And then here's the mucosa. So it's very different. Keep in mind, if you could look for both composition and also functional gut microbiome when you're doing research and develop your products. So this slide shows another example, even at the same site. Let's say we talk about a large intestine. And then when you look at the microbiomes at the mucosa level and also at the lumen level, when you compare to the fecal levels, they are microbiome profile is very, very different. So this is a human study published in 2018. And this part showing where they took the sample, stomach or the large intestine, this is the site they took the samples. And then look at the up left panel, this is a heat map of the microbiome relative abundance of genera, the bacteria in the mucosa, in the lumen, this is lumen, this is mucosa, and this is the stool. And you can see they are very, very different. So the stool sample is not a good predictor of mucosa microbiome. This is something we have to keep in mind. Using a stool sample have some limitations. And this is the pathway between the mucosa and lumen and the stool. So this I'll call it the functional microbiome. You can see this heat map also show the clear difference between mucosa and between stool. Again, the stool is not an ideal or good predictor of mucosa microbiomes. So keep those in mind when you're doing research. Now let's move to dog and cats. Our favorable. So this study published last year, very nice study to look client-owned dog and cats. They do the microbiome, gut microbiome study, and they use the fecal samples and they use the 16S technology measure the microbiome, gut microbiomes, okay? And this slide A, so on the left side, you can clearly see dog and the cat's gut microbiome are very different, okay? If you look at from the higher level, I mean, phyllis levels, and those different relatively small. But if you go deeper, higher resolution to the species levels, and you can see the difference become bigger and bigger. And the similar things you can see on this right side of the figures, it's showing the difference between cats and the dog, and this line, and also dog. So even within the dog, same species, but individual have a difference. There's a big difference between individual dogs, and the similar things for the cats. There's a microbiome, gut microbiome are different between individual cats as well. And again, if you look at the higher level, fellow level, the difference is small. But when you go to the species level, and this is about the species level, you can see the difference become much bigger, okay? So that gave you the higher resolution to look at the gut microbiome in dog, in cats, and also between species. And this is the same study of the previous slide. You can see here, this is the cats, this is the texta, about 83 textas here. You can see the composition is very, very different. The relative abundance also very, very different. Looks like a cat has a more rich in bacterial species and also abundance of those bacterial species. Cat dog is relatively less, but you can clearly see that dog and cat's difference here in their gut microbiome. And this is measuring the bacteria again. And this study also have an interesting finding, says the dominant phyla in dog and cats are similar. So they are the same. There's a 3 dominant phyla here is the Firmicutes, Proteobacterius, and the Bacteroides. So this is the 3 dominant. But if you read other researchers, you may find additional dominant phyla there. But this study using the client on the dog and the cats and found this the 3 dominant phylas in dog and cats gut microbiome. It's very interesting. Now, for us to understand the gut microbiome, what we are more interested want to know is how do we manipulate gut microbiome so that the gut microbiome will promote the pet health. There's many ways you can manipulate the gut microbiome. Of course, diet, this is a gut. We have to eat food, and the dog have to eat their food, the cats eat their food. So diet, the ingredient, any component in the dog food, the cat food has a potential to influence the gut microbiome. Of course, prebiotics and probiotics and also postbiotics, those are very useful tools to manipulate the gut microbiome. Of course, you can use antibiotics that will kill certain bacteria, allow other bacteria to grow. They have the antibiotic resistance. And then you can do the fecal transplantation. Take a fecal sample from one animal and put into another animal. And this is also true for humans. The antibiotics and the fecal transplantation are very often used by the medical field, veterinary medicine or human medicine, not very often used in the pet food industry. So that's why today, let's focus on the top 2 points, how the diet can affect the gut microbiomes and how the prebiotics, postbiotics, and the probiotics, how they can affect the gut microbiomes. And this study published last year, this study looked for the diet effect on the gut microbiomes, and they tested 3 different types of pet food. You can see here is the dry pet food, this is the raw pet food, the dog food, and this the homemade pet food. So 3 types of different kind of pet food. And they feed to dog and then look at the gut microbiome at the phyla levels, at the family level, and at the genus level. And you can clearly see the diet type can affect the gut microbiome. So this yellow is the homemade, is the raw diet. And this blue is the homemade diet. And the green is the typical dry kibble, dry dog food. You can see it. Food type, I mean, can affect the gut microbiome. So this also a study published last year and look at the different protein level in the dog food on the gut microbiome and also the metabolomic in urines, in cereals, and in feces. The protein level they use in the dog food is 47%, 25%, and 19% about. Okay, and then on here you can see the gut microbiome. You know, the bacteria's relative abundance when feed the different level of proteins, and they changed. They are different. And on the functional microbiome, gut microbiome level, if you look at the feces here, and they also affect the function microbiome part. And then these days, you will see some scientists emphasize those metabolites really come into the blood, and that can play their role. That's their opinions. So even for the serums, you can see with the different level of proteins can affect their metabolomics in the serums. Now let's talk about the prebiotics. What is the prebiotics? ISAP has a definition. ISAP stands for International Scientific Association for Probiotics and Prebiotics. So their definition is a substance that is selectively utilized by host microorganism, conferring a health benefit actually to the host. So here's the key point: is something that utilize the microorganism, the host microorganism, but that provide a health benefit to the host, okay? And I think people in the pet food industry or doing pet nutrition are very aware of the prebiotics. And there's a lot of prebiotic currently used in the pet food. For example, FOS, MOS, this is the yeast, most of the times used in the yeast cell wall, inulins, chicory root, beta-glucans, and also the beta pulp. This is very, very commonly used in the pet food. So this prebiotics clearly can affect the gut microbiome because it's selectively utilized by the microorganism in the host. Okay, so that is very clear. Prebiotics will affect the gut microbiome. Now, what about the probiotics? The most commonly used definition is FAO and WHO, the definition for probiotics. So the definition is a live microorganism. I emphasize here is a live microorganism because it has a lot of implications there, which when administered in adequate amount, this is very critical. You need to have to eat enough and then confer a health benefit that you can call it probiotics. Okay. Again, oftentimes we're talking about bacteria, and then here we let's understand the nomenclatures. Oftentimes you will see the bacteria probiotics has a very long name. In this case, is this bacteria is, let's say, Bifidobacterius animalis CECT 8145. So this composes three parts. The first part usually is the genesis describing the bacteria, and the middle part is the species, and the last part is the strain. Most of scientists will say the health benefit is strain specific. So it's not any strain under the same species can provide a health benefit. So even under same species, if they are different strain, you have to test each strain to make sure they can provide the health benefit. Then you can call it the probiotics. And then when we talk about the probiotic, because this is a live organism, we have to think about the safeties and also the efficacy and the stability. For the safety part, you know, this is a microorganism. They could carry antibiotic resistant genes. They could carry a gene that produce toxins. So that's bad. So you have to make sure this microorganism are safe. FAO and WHO has a guideline to evaluate the safety of probiotics, and that is developed for human use. But that principle can also apply to the pet. You know, when we evaluate a probiotic for pet, we can use the same guideline principally. The efficacy part I already mentioned is a strain specific. So you have to clearly identify what strain, and then you can see if that particular strain can provide health benefit to the host. And the host have to be target animal species. In our case, we talk about dog and cats. So if this strain has a health benefit to human, doesn't mean it automatically also have a health benefit to dog and cats. So we have to make sure it has a benefit to dog and cats in our case. And because it's a live organism, microorganism, we have to think about stability, the processing, the storage, the GI system. Pet food making using very harsh processing condition, extrusion, retort. And the purpose of retort is to kill the bacteria, kill the microorganism. So we have to make sure the probiotic can survive the process. And also pet food typically have a long storage shelf life. Typically dry food will have 12 to 18 months shelf life. So make sure the probiotic can survive the long-term storage. And usually we talk about the probiotics looking their function in the large intestine. So they have to survive the low pH in the stomach and also the bile in the small intestine so they can reach their target place and to play their role. So make sure all this aspect you consider when the supplier go into you selling their probiotic, quote, probiotics, and ask those information when you do your own research and think about those aspects as well. So what about the regulations? If you look the regulation in US and Europe, there's no clear quote probiotics in the regulations. In US, AFCO have a direct-fed microorganism. So they don't call it a probiotics, it's just a live microorganism. There's a 12 genesis in the 49 species. So 5 of those are not allowed for dog and cats. And AFCO doesn't go to detail as to strain level, so only to the species level. So there's no strain information in AFCO. In EU, it's pretty simple, about 3 species, strains there, 3 strains there. You can see here, the first 2 looks very, very similar, but that's the 3 strains allowed in dog food and cat food and supplement in Europe. It's called the gut flora stabilizer. It's not called a probiotic. So be careful when you use probiotics in your labels because you have to demonstrate it's meet the definition of probiotics. But pet food industry in US and in Europe use those live microorganisms, I think, is to take advantage of their potential health benefit, potentially used as probiotics. How those live microorganisms in US is labeled. You can see this is an example ingredient statement, and here's how they label the live microorganism. Basically, it's called the dried bacteria's name and the fermentation product, and that's the way it's labeled. Pet food can have a single bacteria or microorganism, or can have multiple species. So in this case, you can see it's the multiple species here used in this pet food. the pet food company can make a GI guaranteed analysis, or they don't make any GIs. In this case, they make a total microorganism guarantee, label guarantee is 100 million CFU per pound. And then pet food may make a claim or may not make a claim. So it's very different in US pet food market. Different company have different approach. Some pet food company do make a claim, typically it's related to their gut health or immune functions. Usually they will have some kind of data research to support their claim. And then postbiotics. This is very, very new. At this moment, there's no agreed definition for postbiotic in the scientific community. However, the ISAP now come up a consensus statement, group of international experts, they all together have a meeting, discussion, finally they come up with this consensus statement, and that they submitted for publication, and I hope that their publication will come out any day soon. Last year, this definition, ISAP definition, was introduced in a conference. So now we can see what ISAP definition is. So for ISAP, postbiotic is called the preparation of inanimate microorganism. And basically, this microorganism is not not alive anymore, and or they are component that confer a health benefit on the host. So in other words, the postbiotic is a deliberately inactivated microbial cells or cell component, either with or without their metabolite, and that confer a health benefit on the host. And you can see here, the postbiotic really compose a lot of stuffs in it. Can be the dead bacteria itself, it's a fragment cell walls, it's a content enzymes, or it's a metabolite. So all those together make up the postbiotic. And you can see clearly here, the postbiotic has a huge advantage over probiotics because now you can have a less concern about the stability. So gut microbiome is a really active, growing very fast research areas, also in the marketplace, also going very fast. You can see a lot of publications with this gut microbiome related to the health, humans and the pets. Those biotics, prebiotics, probiotics, postbiotics, has a significant effect or role we can leverage to manipulate the gut microbiome to make it more healthy and then to promote the pet health. And you probably also heard of symbiotics, parabiotics. So it's a very complex, lot of stuff. But fundamentally, I think there's three prebiotics, probiotics, and postbiotics. Three biotics has a major role we can use to innovate, to create better formulas to promote a healthy gut microbiome, and then have a great opportunity to have new product innovations. So in a summary, today's talk, we covered the microbiome is a growing area, both in market and in research, and hopefully that will improve the pet health. And also gut microbiome, typically we use fecal samples and the high throughput technology. And those technologies and samples is convenient, is very useful, but also have their limitation, particularly using fecal sample to predict the gut microbiome. There's some limitation, we talk about that. And then we also talk about the tools we can use to manipulate gut microbiome, for example, using diet, particular those biotics. So I'm expecting more research and more studies and even more pet food that can support health microbiome in pets in the near future, probably in the next 3 or 5 years, you probably will see more of those innovations. Thank you very much for your attention.
Muchas gracias. Thank you very much, Dr. Yu. Vamos a pasar a las preguntas y respuestas. Here's one from Fernando R. Feucher. He said, well, congratulations, very good English and very clear accent. And he's saying that as a child 50 years ago, very different dogs ate anything from our home table, some homemade food for dogs. They were healthy and strong. What is happening today? The dogs eat processed pet food from the grocery, and now they easily get sick if they eat something different from the dog food bag. Can you please explain us what are the reasons about this, Dr. Yu?
Oh, thank you very much. That's a very challenging question. I don't know I can give you a good answer, but here's my my thinking. You know, those kind of individual observation versus a larger real study probably is needed to answer that question. I mean, if you look at the longevity studies 20 years ago or 40 years ago, when dogs just eat homemade food or whatever, you know, left over from the table, or they go wild to catch animals. Compared to the longevity, these days, home pet dog and cats live much longer than 20, 30 years ago. Of course, that's not just reflected by food itself, but also reflected by the advance of the veterinary medicine and the medical care. But I don't have a good answer to draw the conclusion that the modern pet food, you know, is bad. So that's probably not true. You know, there's so many different pet food on the marketplace, and the quality in nutrition-wise, the quality is very, very much different. You just cannot just read the labels because some pet food play ingredient rather than nutrition, but you also have to look at the nutrition, all those things, and then you can see if this modern commercial food is really bad. So that's my thought.
Thank you very much. Good thoughts. Buen punto. Ricardo Basurto, in the study of IBD versus healthy dogs, the changes in bacterial populations, could it be explained by types or amounts of food ingested?
No, that study, they control the food. So all dogs eat the same food, and then they look at the microbiome. But now again, IBD is a very complex disease. And at this moment, we don't know what's the cause of that condition. And then if you look at the microbiome change, because it's a highly inflamed large intestine there with the IBDs, so it's the egg and the chicken question as well. This is just one of the observation study to look at, you know, the microbiome are different. But let's say if you have a magical way to make those gut microbiome looks like a healthy dog, will that help to, you know, treat the IBD? I think that's a good question. I remember Texas A&M group, you know, they have a very strong experience doing this kind of research. They did a study to look at the probiotics treat IBD, and if you look at the publications, they do see some improvement with that treatment, and also change the gut microbiomes, but it's not you know, 100% cure the IBD. So I think this is a very complex question.
Okay, completely agree with you. Thank you very much. Ricardo Yescas, what is your opinion about sodium butyrate? Be it coated or not? Thank you. And I will say, make that question. Yeah, I mean, is it advisable to use to change the pH? It's just for puppies, for adults, for seniors. What's your opinion in general about acidification, specifically in this case, sodium butyrate and coated sodium butyrate, please?
That's yeah, it's clearly so this is related to the company, so I have to be careful to comment on this. But that ingredient I used it before when I worked for a company, when I do the formulations for global pet products. I do see that products. I did use that product before. It clearly can reduce the urine pH in both dog and cats. So in that part, it's effective. Okay.
Yes.
But then there's some handling challenge because it's a very attracting moisture. So in the manufacturing facilities, let's say in some part of Mexico, could be high humidity in the plant, and the handling is some challenge. But to reduce urine pH, it is effective.
Yes, and I don't know Ricardo Yesca's idea or approach of the question, but basically, okay, about the use of acidification in terms of urine, that's perfect. But in terms of helping to make a better environment for the gastrointestinal tract, what's your opinion? I mean, using it or not? It's like, for modifying the pH of the gut.
I haven't read any data, see any publication to see how that compound can modify the gut pH, because it modifies the urine pH through metabolic way. So it's directly put there to change the urine pH. It's through the metabolism, change the pH. It's just like when you gave methionines and when you gave other acidified agent, it's through the metabolism and then end up in the urine and then make the urine acidic. But in the gut, with the amount you add in, it's probably already absorbed into the body rather than reach the colon, reach the large intestine to affect the bacterial population there. This is just my pure guess, theoretical thinking, but I haven't seen any data to show that will change the, gut microbiome. When we use prebiotics to lower the gut, pH, it's basically, we are trying to use those prebiotics to encourage those acid-producing bacteria growing. Typically we call the good bacteria, good bugs, bifidobacteria. They produce acid, like the particular those short-chain fatty acid. That's make the fecal samples pH reduced. But if you use those compounds, if that will reduce the fecal pH or the content of larger intestine, I haven't seen any data yet.
Wonderful, wonderful. Thank you very much for this answer. Ricardo Rodriguez makes another question. Considering the given definition of postbiotic and being this very recent definition, the process of manufacturing of extrusion can affect these postbiotics. Is there any supplier that has developed these postbiotics that can be added to the pet food? I mean, without having any change or problem or a kind of problem with the extrusion processing?
That's a very good question. As I mentioned, the postbiotic is a relatively new areas. You can see more publication on the human side. ADM is very active in those areas. If you read the recent publications we made last year and also early this year, we are looking one probiotics. Commercial name we call the BPL1. We look at in not a dog and cat yet. So we look at in the lower species is called the C. elegans in that model. Mm-hmm. We look at the rats models, and we looked at human data, human studies. In those studies, we clearly can see live bacteria probiotics and also heat treated. So we kill those bacteria and feed it to C. elegans and feed it to rats and also to human clinical trials. And this heat treated, let's call it the probiotics, have the same effect to manage the metabolic syndrome. So those obese, overweight people have a metabolic syndrome. And this heat-treated version, probiotic, have that same effect. So I'm currently running a dog and cat study for your information to test if that also true in dog and cats. So if it's true, I think that's a good advantage to use in the pet food. Now, there's another question we have to consider is the extrusion and also the retort. Even though the postbiotic is killed bacteria, so they are not alive anymore, but those active benefit compound, will they also survive the extrusion, also the retort? I don't have those information yet, so I think that's a good question. We need to look into it. But at the least, if we apply that post-extrusion for the dry pet food, and I think we have more confidence that the postbiotic will work, because a lot of the probiotics we applied is after post-extrusion, and then particularly those some spore-forming probiotics, and they are very stable during the storage as well. When they come into the animal body, they can regerminate and become live and then can provide health benefit. So from that perspective, I think a lot of those postbiotic has a great potential.
Wonderful, thank you very much. Y simplemente ahí agregando, estoy totalmente de acuerdo con el doctor, tratar de privilegiar el uso post-extrusión. O sea, no queremos ni la presión, ni la humedad, ni el calor de la extrusión, ni el tiempo y la temperatura del secado. Entonces, lo más fácil es tenerlo aplicarlo al final cuando se aplica la grasa y el saborizante. El doctor José Luis Laparra, gusto en saludarlo, tiene una pregunta. Dr. Laparra asks, please tell us the mode of action of probiotics with regard to organic acid production. Is this why probiotics are functional? What about administering directly organic acids, mainly butyric acid, to the food to be consumed by the animal to obtain the benefits more directly?
That's a very good question. I think that will take $1 billion to answer. So from a very simple way, there's a lot of hypotheses, theories, how those probiotics works, but I don't think we have a clear understanding, agreement of what's the mode of action. And there's a lot of hypothesis theories that it could be those probiotics will have some metabolites. They produce, GABAs, those neurotransmitters, and they produce some compounds that will affect the immune systems. They have their own, let's say, metabolite. Let's say some bacteria will produce vitamins that is critical for the animal health. A lot of hypotheses the way it could probiotics work to improve the health. But then you talk about each different probiotics, each different species, different strains, and they may also work differently. Some produce acid, like say short-chain fatty acid, and then some may not produce that short-chain fatty acid. They just produce some small molecule that used as a neurotransmitter and affect the whole physiology of the animal. So that's a big question. I don't think we have a clear answer, understanding yet. And then for that postbiotic, if I look at the ISAP, they clearly state that if you just isolate one of the components, for example, you say it's a short-chain fatty acid, butyrate, for example, butyrate, and then you say that is a postbiotics and then provide help. And from from the ISAP's point of view, that's not considered the probiotics. So if you just have one simple compound, that may have some health benefit. I mean, if you feed that, if you protect it, not absorbed in the small intestine, can reach the hindguard, large intestine. I mean, butyrate can be helpful, provide a certain health benefit because of energy for the large intestine cell. I do see some commercial product available. They have kind of microencapsulated forms, so they can protect it to make sure it reach the large intestine. That is used in the livestock industries, and it could be in the pet food as well. Again, I don't think we have an answer. What's the mode of action of those probiotics?
Wonderful. Thank you very much. Patricia Pauls. Hola, Patti. Does grain-free dog food should be supplemented with some probiotics always?
I mean, grain-free is, you see the FDA, now it's a little bit quiet, but, you know, let me put this way. Grain-free product is about ingredient. It's not about the nutrition, okay? So as long as it has a sound nutrition, it should not make a much different from other pet food. So all the ingredients may contain all different compounds in addition to the nutrient. They may play some biological role, but it's not a specific, grain-free. I mean, even in the grain-free, you will see different companies use a different ingredient. So subcategory-wise, they may also different. So in my opinion, it's not grain-free or non-grain-free regular dog food that use in the probiotic. You can use the probiotic in any kind of pet food as long as this probiotics survive, because this life and microorganism can survive the process, survive the storage, survive the GI system, reach their target in the site so they can play their role. I don't think it's have to be grain-free.
Wonderful. Thank you very much. Another question from Ricardo Yescas. He says, I'm thinking about controlling potential pathogens. Will it work? I don't know what is he referring. It's like probiotics maybe, but the point is controlling potential pathogens.
It is. I mean, there's a good evidence to support certain probiotics can compete with the pathogens in the gut, prevent those pathogen bacteria to adhere to the gut, cell. So this is one of the mechanisms how the probiotic work. So one of them. So there's some good evidence can show in that the probiotic do compete with the pathogens by numbers, by binding site. This is a mechanism people talk about.
Perfect. Mónica Alejandra Mendoza, do you have any experience with any type of biotic as part of a treatment of a bacterial infection disease as principal response without the use of antibiotics? Also, can you share your result or opinion if you do?
I do not have my own study, but you do see some publications, particularly those acute diarrhea in dog, using probiotics can reduce the course. Let's say if you don't use antibiotics, let's say if it's a bacterial infections, if you don't use antibiotics, you don't use probiotics, let's say they last for 7 days or 8 days. But if you use antibiotics, they can shorten the course. Let's say they will recover in 3 days. You do see those publications with the probiotic can shorten the course of acute diarrhea. And that's an example of, let's say, intestinal infections. Let's say IBD is kind of infection as well, but may not be bacterial caused, maybe something else caused. but it is an inflammation. So there's some kind of infection, I think, involved in the process as well. Texas A&M study using the probiotic do help a little bit as well.
Perfect. Thank you, Dr. Yu. Here comes a very, very nice and interesting question. It's El Bautista. Is it possible to manipulate the microbiome using different fiber sources? What kind of fiber fractions should be used for increased healthy microbiome?
That's a very specific question, a very interesting question. I think that's a fall into the prebiotics. Because if you look at the prebiotics, we're looking for most of things, the polymers, okay, is from the plant source. It's basically fiber. A lot of those prebiotics are soluble fiber. But now if you ask, okay, if I use FOS, okay, for that particular prebiotic, those particular fibers, and then this also involves the length of the chain. Is it 2 polymers? It's 11 polymers, or even come to the inulin levels, or come to the chicory root levels. So that could be the different fraction of the fiber if you can. But which fiber cause which bacteria grow? That's a big question. And there's some study to show, let's say, if you look a lot of this in vitro fermentation study conducted by the University of Illinois, Maria's and Kelly's and George, and they did a lot of those kind of work. They do so certain fiber type, promote a certain bacterial growth, inhibit the other bacterial growth. But I think when you come to that level, specific, you have to do those kind of research to find out.
Perfect. Thank you very much. Now we go to William Quevedo's questions. What's your opinion of the combination of xylanasas, hylanasas, I don't know what's the right translation, and prebiotics to promote intestinal health? It's an enzyme from xylose. Okay, did you get my point or
sorry,
not?
Sorry, I don't know. But if you mention it's an enzyme.
Yes, an enzyme from xylose, xylanase, and probiotics, both together to promote intestinal health.
I think that's a good question. That's a good question. Enzyme in pet food application has been discussed a long time. Will that enzyme help, just like livestock? Livestock use a lot of enzyme, but we don't use enzyme very much in pet food. Oh, it's a xylanase. Thanks, Armando. Yeah. So that enzyme is to break down the fibers. So in particular in the ruminant, and then the enzyme will help the energy utilization. But if you use that enzyme to break down the fiber to have there's a prebiotics we call the XOS. I think XOS. That's if you use that enzyme, will come up with that kind of prebiotics. But enzyme itself is not a prebiotic. I don't think enzyme itself is a prebiotic. But if that enzyme work on the fibers, that will could produce a prebiotic that will promote a certain health benefit. Certain good bugs to grow, inhibit the bad bugs. So that could provide a health benefit. But that will become more complex. You think about enzyme typically need a condition to work. If you think about will that work in the stomach? I don't think so. It is a very low pH, probably not favorable for the enzyme activity. So when they come to the small intestines, they may work, and hopefully they work in the large intestine environment. The enzyme will work. And then you have to provide enough substrate to allow the enzyme work to produce sufficient, quote, unquote, prebiotics to improve the health of the gut microbiome. I think it's very complicated. If you are interested, you can run the study to see, your strategy, combine those together, and then you can measure the microbiome, see if you can achieve your goal.
Thank you very much. Another question from William Quevedo. What's your opinion about the use of animal plasma in pet food? Are there any benefits?
I think plasma is a good protein source. There may be some biological active compound in that material as well. So I don't recall exactly, I do see the studies to evaluate the animal plasma as a protein source for pets. Is more focused on the digestibilities, amino acid profiles. I haven't seen any good study to show additional benefit, but it's a high value protein source for pet dog and cats, so that's true.
Thank you very much. Sí, también exactamente. Igual he visto varios estudios y prácticamente el enfoque es hacia digestibilidad y hacia valor biológico de la proteína, que es muy alto. The last question from William Quevedo. Do you think that the use of yeasts in pet food can activate the immunological system, and therefore will there be any expenditure in nutrients because this task?
I mean, yeast is pretty widely used in the pet food, and in particular in cat food as a pelletant to improve the palatability of cat food. But yeast also used as a functional ingredient to improve the gut health by improved immune function, gut immune function. Particularly when you break down the yeast to extract the yeast cell wall that have a beta glucans and other component. And then I think Alltech did a very nice product development. They further, disaggregate yeast cell wall and get different functional component. And some of them have a very nice immune function, functionality, improved immune function. Remember, gut is the big immune organ. So a lot of immune function is along the intestine wall. So that's why yeast cell wall and the yeast, they all provide this kind of functionality to improve or stimulate the immune function. If you look at one of those prebiotics, we call the MOS, a lot of those products actually come from the yeast cell wall, and very typically you can see Alltech have that products. So yeast do provide a health benefit in that regard.
Perfect, thank you very much. Another question from Alberto Miranda. In feedstock, there's been an association between the profile of the microbiome, the ruminal microbiome, and the productive efficiency. How much or what is the influence of the genetics of the animal in selecting the microbiome that will live inside that animal, and how much the microbiome in the efficient animals is the result of the diet that was consumed in the early stages of the animal's life?
Now that's a very great question. Ruminant is a completely different game. So I'm not specialized in ruminant, but do know that the ruminant is a very critical organ for the health of a ruminant. But this is very, very different from dog and the cats. So they are single monosystemic animal. The system is very, very different functionally. But good question is there's some gut microbiome. They probably play a similar way, improve the health, because a lot of research of this gut organ access and the gut microbiome associated with different disease and conditions. And I didn't talk about that, but if you look at the publications, gut microbiome is very different at different age stage. in the puppies, in the early adulthood, in the adult, and in the later life senior, their gut microbiome is age-related. And this is similar in humans. A lot of study done to measure the human gut microbiome associated with age. And in humans, different way of deliver a baby can also have the effect on the gut microbiome. Naturally deliver or C-sections, and their gut microbiome in the early life are different as well. So that is a great question. So gut microbiome is very complex, and then let's also rate individual specifics. Some individual microbiomes are very, very different from others. And in humans, there's a study to look at the probiotics, gave probiotics, and then see how this probiotics will stay in the, colonize in the large intestine in the mucosa, because that's the place we believe that will play a role. That study found that you basically can divide people into 3 different categories. One, their microbiome at the mucosa level are resistant to any probiotic. So if you gave the probiotic, it doesn't have any effect. But the other ones, they are kind of open. They are permissible. So they allow those probiotics to colonize in the mucosa. And then the category is somewhere between. They can colonize in a transition period, but then they are gone. They all disappear. So very, very individualized. So I think gut microbiome or microbiome itself is a still big mystery. A lot of research needs to be done to truly understand it.
Wonderful. Thank you very much. Here we have another question from Ruben Ortiz. Which ones are the 5 probiotic species that are not allowed in dogs or cats?
That's a good question. I think nutritionally, I'm not aware of any particular prebiotics not allowed in dog and cats. In general, most of the countries, regions I'm aware of, there's no specific limitations for prebiotics. But maybe some ingredient for certain countries is not allowed. For example, that human milk oligosaccharide, that particular prebiotics is used in the human food and the supplement drink. But I'm not sure is that approved ingredient for pet or dog and cats in US or EU. I haven't checked that yet, but there could be some ingredient and that's not approved for dog and cats in certain region or certain country. So you have to check which market you play, check with the local regulatory agencies to make sure the ingredients you use are allowed, approved for the pet food and dog and cats.
Thank you, Dr. Yu. Another question from Ligia Jaimes. Well, not before Ligia Jaimes, I got one from Francisco Guerrero. How convenient is the use of soybean husk or soybean hulls or any alternative fiber raw materials to manipulate the content of the intestinal microbiota, and what is the limit of its inclusion?
That's a great question. Soybean hull, I think in last Amina, Gary presented a great study on soybean hulls on the digestibilities. In that study also have a microbiome analysis. So in that study, if I remember it correctly, the recommended up level is 10% in pet food. So you can go higher and lower, but in that study, I think they used 10% soybean hull in the study to show the results is comparable to beet pulp. Yeah, in regard to their functionalities, and then also the gut microbiomes with a lower cost. But that's a study I saw more systematically and research at the soybean hull. I haven't seen other study doing that at the detailed research.
Correct. Yeah, perfect. Thank you very much. Vamos a hacer por términos de tiempo una pregunta más, ahora sí que en vivo, no hay ningún problema. Recuerden que las preguntas las tenemos documentadas y se las damos a Dr. Yu para que nos la conteste. No se van a quedar preguntas sin resolver, nada más en vivo vamos a hacer una pregunta más y las demás, insisto, se las vamos a contestar. I'm telling them that we're just going to do another question and the rest of them will be sent to you so you can answer them afterwards, okay?
All right, thank you.
So the next and the last question is from Ligia Jaimes. What is your recommendation with phytobiotics? They are medicine plant extracts, and what's the relationship with the microbiome in pets? It's like mixing or using these phytobiotics in regards in the microbiome of the pets.
Sorry, can you say that question again?
Yes, it is referring to phytobiotics, the plant extracts, medicine plant extracts, the relationship between these phytobiotics and the microbiome in pets. I don't know if you heard about this, or you have information.
Let me see if I understand it correctly. You're talking about the fecal microbiome or fecal transplantation.
It's like plant extracts, plant extracts like, I don't know, from oregano and several plant extracts.
Okay, so the plant extract, extract. Sorry. Plant extract on the gut microbiome.
That is correct.
I think there's good potential. There's a possibility. You think about that plant extract can be very complex, and depending on how you extract it, which plant you do the extractions, and then the profile of different compound in that extract could be very, very different. And then will that affect the gut microbiome? Remember, we talked about the gut microbiome. The major piece is the microbiota. A lot of different microorganisms there. And, will that affect that? I think there's a good chance those extracts will affect the gut microbiome. But is it to make it in a good way or in a not desired way? I haven't seen any good publications, so I not know that. But there may be already some publication out there, but I haven't do the systematic research. Survey to see how that will affect. So no, I do not have good comments on that.
Thank you very much. Well. Saying that, Dr. Yu, I appreciate very much your participation, your knowledge, and all this wonderful information that you share with us this evening. I guess we're finishing the questions and answers, your presentation, and we will leave the communication to Ana, Dr. Guerrero, and Jorge Cervantes. Thank you very much, y muchísimas gracias también por haberme invitado a estar aquí con ustedes.
Well, thank you very much, Miguel.
Thank you. Ana.
Perfecto, muchas gracias, Miguel. Muchas gracias primeramente por tu trabajo como moderador. Gracias por acompañarnos por aquí con tu experiencia también en este tema de salud intestinal de mascotas. And we want to thank you, Dr. Shiguang Yu, for being here with us with all your knowledge and taking the time to answer all these questions.
My pleasure.
Thank you. Igualmente, pues agradecer a la empresa ADM por ayudarnos con este seminario. Y les puse ahí en en el chat una serie de enlaces. Hay un enlace para una encuesta de este seminario, si nos pudieran ayudar contestando la encuesta. Es súper sencilla, nada más es para darnos una idea de su opinión de nuestros seminarios y hacia qué dirección dirigirlos. Igualmente les presento aquí el cartel de nuestro próximo seminario. Este es un seminario de la industria. Es el próximo martes 9 de marzo a las 17 horas. Vamos a tener el tema con temas de avicultura, cómo enfrenta la avicultura brasileña la prohibición del uso de antibióticos y estrategias para mejorar la salud intestinal en aves y la producción sin antibióticos. Los esperamos entonces el martes 9 de marzo. Igual ahí en el chat les dejé el enlace para que se vayan registrando. Dentro de 2 semanas, el martes 16 de marzo, tenemos nuestro 5º seminario de nutrición. Este es alimentación avícola sustentable. Nuestro ponente es el doctor Sergio Fernández. Va a ser el martes 16 de marzo a las 17 horas. Igual ahí en el chat está el enlace para que se vayan registrando. Y pues les vamos pidiendo que aparten las fechas de nuestros siguientes seminarios de la industria, el martes 23 de marzo, todos son a las 17 horas. Entonces, martes 23 de marzo, martes 6 de abril, martes 20 de abril, y por ahí les estaremos mandando igual las invitaciones de todo esto. Igualmente, pues hacer de su conocimiento que tenemos nuevas fechas disponibles para los seminarios de la industria. Estas son 4 y 18 de mayo, 1, 15 y 29 de junio, 13 y 27 de julio, 10 y 24 de agosto. Ya saben, si están interesados, es cuestión de que me envíen un correo de administración @mena.mx y podemos ir apartando sus fechas. Y para terminar, pues les dejo el enlace también por ahí de nuestro canal de YouTube de la MENE. Están las grabaciones de nuestros seminarios anteriores, estará este también. Y pues nada más, ¿no? Desearles una excelente tarde, agradecerle nuevamente a Miguel por su trabajo de moderador. Thank you once again, Shiguang Yu. Have a good evening. Y
Thank you, Anita.
Y nos vemos entonces la próxima semana, el martes. Gracias a todos. Thank you all. Gracias a Lulu también, a Lourdes Rivera por la traducción.